All right, so today we're going to talk about metabolism, and the textbook references for metabolism are actually in chapter 24. So we kind of bounce to the end of the book, and specific page numbers in there are starting on page 934 in your textbook, talking about metabolism and anabolism and catabolism, and goes through pretty much the end of the chapter. So that's your page reference for that. So the reason we study metabolism is we know that there's different factors that affect metabolism, those chemical reactions. So defining the word metabolism, it's the sum of all the chemical reactions in the body. So when you think about, making glucose between meals versus breaking down glucose after meals, and the production of hormones, everything that's occurring in the body is summarized as this term metabolism. So some people say, oh, I have a really high metabolism, and that means when they're building and using energy for metabolism, they're using stored calories or calories from food they've just eaten that might be still circulating in the blood. They're using that for energy. And other people with slow metabolisms, it takes them longer to break down those products, so they're more likely to store excess calories as fat, and then as a result they have more trouble with their weight. And that can change throughout the lifespan. We know that, like in women, the role of estrogen affects metabolism. So women find when they get to the end of their reproductive years and estrogen levels are declining, they start to have fat kind of developing around the midsection, versus earlier in life, maybe they didn't have that kind of issue. So another thing is, as we age, we tend to slow down and be less active, and then people gain weight because of that. So it's not always, you hear people say, oh, I had kids, and that's why I'm 40 pounds overweight, or oh, I'm older, that's why I'm 40 pounds overweight. It has a lot to do with activity. Is when women have children, they're more focusing on the kids and less focusing on exercise or their diet, and they tend to eat kid-friendly foods. I mean, I'm guilty of it myself. I'll try to be watching when I eat, and then I'm making the kids macaroni and cheese, and I'm thinking, oh, that looks really good. So it's a challenge when you have children to try to eat healthy. So the best way to do that is obviously cook healthy things for your kids, right? And then eat right alongside them. We don't have to feed our kids these kid-friendly foods, because what are we doing? What are we teaching those kids when their diet is peanut butter and jelly, pizza, and mac and cheese every day? What are they going to do when they grow up, right? When they're teens, they're going to continue to eat those kinds of foods, and they haven't adjusted their taste buds to vegetables and fresh, fruits and things like that. Whole wheat breads. If they've never tasted white bread or was very rarely served white bread, they're not going to crave white bread. Well, maybe they will if they like the taste of it, but they're going to be used to whole wheat bread, and they'll tolerate it. And it's a real problem. Sometimes my kids have play dates, and their friends, all they want is pasta with no sauce and bread and just really low nutrient, laden foods. So we have to be careful with that and make sure, you're encouraging your kids to eat a variety of foods and getting those taste buds used to those foods early on. So it's all about that. I mean, when you think about metabolism, the importance of understanding metabolism is understanding what we put into our body and how it's processed to produce all the different molecules we need. And certain molecules are better at being converted to energy than others. So it helps us make decisions on what foods are best to offer our patients and also ourselves. So two different chemical reactions involved with metabolism. There's the building up and the breaking down, and that involves either the release of energy or the storage of energy. So when you look at anabolism, anabolism is building molecules in cells. So when we talk about protein synthesis, that's a classic example of anabolism. When we're making proteins, we're building those large molecules from smaller ones. What are the small molecules are using? Amino acids. So we're linking those together, forming peptide bonds. The formation of peptide bonds stores energy. So when we break down protein, when we break bonds, we release energy. So anabolism is the storage of energy for later usage by the body. So when we are building fat, excess glucose and calories from our diets, if we're not using it, we store it. The body is very selfish and doesn't want to get rid of excess calories, so we store it as fat. And then catabolism is the opposite of that. It's breaking things down. So digestive enzymes in the digestive tract, that's a classic example of catabolism, because we're taking the proteins from the peanut in your Snicker bar and you're breaking it down to amino acids. So that's releasing energy when we're breaking those bonds. And that's when you get hungry and tired and hangry when you eat, then you feel better, right? Because you've released the energy from those foods and it's fueling your brain cells, and then you feel better. So catabolism is the release of energy by breaking down larger molecules into smaller. So when I think of catabolism, think of cutting down large molecules and releasing it. And when I think of anabolism, I think of adding to molecules and making bigger ones. So anabolism adds, so it stores energy. Catabolism cuts them apart, breaks them down, and releases energy. So we need a nice balance of both. Of when we bring in our food, we need to digest it with catabolism and then build new things and have a nice balance of the two and have a healthy weight and good energy levels. So when you look at what the key molecule that we're working toward when we talk about energy in the body, and that's ATP. Adenosine, so when we take adenine, which is a nitrogen base, right, and we bind it to ribose, it forms adenosine. And then we bond 3 phosphate groups to it. That's where the tri comes from. So those last 2 bonds that are shown in red here, here and here, those are special high energy bonds that when we break that bond and release it from ATP, we release energy. And then we can take that phosphate that we removed and we can add that phosphate to a molecule, and that gives that molecule energy. Let's say it's the sodium-potassium pump, for example. And that's what drives the sodium-potassium pump to allow it to pump things against its concentration gradient, sodium and potassium, is because we add a phosphate. We phosphorylate that protein, which is the sodium-potassium pump. So that releases energy when we take that phosphate off. And now we have a DP, diphosphate, because we have 2 phosphates on it. We can release another phosphate from that to make adenosine monophosphate. And then we can go the other direction. We can add phosphates to it if we have excess energy in the body, and we can store ATP. So the goal of what we're going to talk about today, when you think of metabolism in terms of making ATP, we're taking our food sources and we're running it through glycolysis, the Krebs cycle, the electron transport chain. Do those terms sound familiar to you? Did you learn about that in general A&P a little bit? Okay, so the goal is to make ATP at the end. So cellular metabolism, when we're making ATP, is that an anabolic or catabolic process? Anabolic, because we're adding phosphates to that ATP. We're building ATP molecules. Okay, and the goal is to have a lot of that, so when we are, driving our cellular processes, we can go to those ATP, stored ATP molecules and get quick energy. So this is a reversible reaction, either making ATP or breaking down ATP. And these bonds are where all the action is, those last 2 bonds on the ATP molecule between the phosphate groups. All right, so cellular respiration. It's breaking down food, so it's catabolic, breaking down the foods we eat. And what are the different types of foods we eat? What are the food categories, major nutrient molecules? We talked about this in the last set of notes. Proteins, fats, carbohydrates, and lastly, DNA and RNA are nucleic acids. So nucleic acids, DNA, RNA. So those are our 4 nutrient molecules: proteins, carbohydrates, fats, and nucleic acids. And we need a variety of those in our diet to fuel metabolism. Some people go to these zero-fat diets. That's really not a good thing because the body needs fat. But it doesn't need it in French fries, right, or blocks of cheese or greasy burgers. We need healthy fats like those found in plant sources, particularly like olive oil is a really good choice. Coconut oil, we know, is a good choice. So there's healthy fats, but we need fats because what are the molecules that make up the plasma membrane? All of our cells are made up of these Phospholipids, yeah, very important fat. So in order to make new cells and support our cell membranes, we got to have some building blocks. So we need to bring in the fats so we can break those down into glycerol and fatty acids, and we can reassemble those into cell membranes. We also know that hormones, some of our hormones are fats. They're lipids, like the sex hormones, testosterone, estrogen, progesterone. Those are fatty hormones, aldosterone. So without those building blocks, we can't get those hormones. Okay, so we're breaking down food fuels, and then we're building up and making ATP. So when we talk about the term phosphorylation, it just means we're taking that phosphate group off the end of ATP and bonding it to another molecule. So it's the direct fuel for our cells, is ATP. So we talk about glucose as being a fuel. Well, glucose is the starting point, but we're trying to make ATP from that glucose, and that's the direct fuel for cells. Just like I talked about in general A&P, the gas for our cars is the fuel that runs our car, right? But I need money, I need cash to buy the fuel. So what does the money represent? The glucose, right? I can't stuff dollar bills, right, into my gas tank. It's not going to work. So the fuel for cells is ATP. We need glucose to get the ATP. We need money to buy the fuel for our cars. So when we look at the stages of just how we process nutrients, stage 1 is digesting. So using digestive enzymes to break those proteins down into amino acids, breaking down our polysaccharides like in your starches, like your breads and your pastas. We break those down into glucose units, small glucose units. And then fats we break down into glycerol and fatty acids, and nucleic acids we break down into nucleotides, right? Because we know those nucleotides are floating around in the cytoplasm for new protein synthesis. So the breakdown products of each of those is shown on page 935 of your textbook. It's figure 24.3. It shows that proteins are broken down to amino acids. Glucose is broken down, or I'm sorry, glycogen is broken down into glucose, and fats in our diet are broken down into glycerol and fatty acids. Okay, so step 2 then, once we've broken down those bits and pieces, now we need to absorb them into, at the end of stage 1 is the absorption into the blood, transporting it to the tissues. And then the cells are going to use those bits and pieces to assemble new things. So we can see stage 2 is synthesis. So this is an anabolic process. We're going to make lipids, proteins, and glycogen from those bits and pieces that we got from our digestive tract. Or we're going to take the glucose and further process it via catabolism, which is glycolysis. And we'll talk about a lot of that today. And then stage 3, we break down some of those products from glycolysis and run them through the Krebs cycle and the electron transport chain, and we make that ATP molecule. So cellular respiration, we think of respiration as breathing, but when it's at the cellular level, we're using oxygen to make ATP, is essentially the sum of those reactions. Okay, so to understand a little bit about these reactions, we have to understand what a redox reaction is, or oxidation reduction. How many of you have heard of that reaction before? Just a couple? Kind of brings back horrible memories from chemistry, maybe. Sometimes it's not very well explained. Well, the good news is it's not a chemistry or a biochemistry class, so we're not going to go into the nitty-gritty details of redox reactions, but I just want you to know some basic things about them. And that is oxidation reactions involve the gain of oxygen or the loss of hydrogen atoms. So it's all about oxygen and hydrogen. And it's all about electrons, because electrons are the energy particle, right, of our atoms. We know that when we lose electrons or share electrons, we form bonds, or gain electrons, we form either ionic or covalent bonds. Covalent if they share, ionic if one loses and one gains. So it's all about the electrons. So if you remember from the water molecule, water is made up of 2 atoms of hydrogen, 1 of oxygen, and we know the relationship of the oxygen and the hydrogens in a water molecule is a little bit selfish, in that water, or in that oxygen kind of steals the electron from hydrogen, and it's called a polar covalent bond. So I'm going to just show a picture here. Trying to find one water molecule. Let's see. We'll look at this one. So if I look at this representation here, here's the oxygen. It's got 2 electrons in its first energy level, and then it has 6 in the outer energy level. How many do they need to be full and complete and stable? 8. So it's going to bond to 2 different hydrogen atoms. Hydrogen has an atomic number of 1, so it has 1 electron in its outer energy level. So if oxygen binds to 2 hydrogen atoms, it can share an electron with hydrogen, and hydrogen needs 2 for its first energy level, so it can share an electron from oxygen. So they kind of hover their outer energy levels over each other, and they share electrons. But oxygen is a little more selfish, which means it pulls those electrons closer to its nucleus than hydrogen can. So hydrogen is slightly negative. So does hydrogen lose energy or gain energy in that relationship with oxygen? Loses it, because it's pulling the electrons closer to oxygen. So oxygen is electron hungry. So because of that, whenever a molecule gains oxygen, that molecule loses energy because oxygen pulls those electrons from that molecule and is selfish, okay? And then when we talk about losing hydrogen atoms, let's see, get the right. So when you lose hydrogen atoms from a molecule, like if you take hydrogen off of a molecule, it takes its electron with it, and that molecule will lose energy. But what if I bind a hydrogen atom? What happens to that molecule if I bind a hydrogen atom? It gains energy because it gains the electron that hydrogen atom has, okay? So again, hydrogen, oxygen are involved in redox reactions. So again, if you look at the notes on page 11, so oxidized substances lose electrons and energy. So an example of that is when it gains oxygen. When a molecule gains oxygen, we say that molecule is oxidized. If a molecule loses a hydrogen atom, we say that it has been oxidized. So gaining oxygen, losing hydrogen means the molecule is oxidized. And the opposite is true for reduction reactions. So reduction reactions, we are losing oxygen or gaining hydrogen. So when one molecule is oxidized, the other molecule that gains the oxygen, or I'm sorry, the molecule that lost the oxygen, we say is reduced, and the molecule that gained the oxygen is oxidized. So these reactions occur in pairs, because it's just the transfer of electrons, right, and energy. So when we talk about metabolism then, the metabolic reactions are all about hydrogen, losing hydrogens, and we're going to see that as we go through the reactions. So another thing that's important to understand is when we talk about this gain or loss of oxygen, hydrogen, it's all about enzymes. Enzymes are catalyzing that process of gain or loss in movement of atoms among molecules. So we need enzymes, and I think that was one of the tricky questions on the prep course, right? Or which of the following are part of, complete, what was it, catabolism, or I forget the term, metabolism of glucose. So we need the ribosome, or I'm sorry, we need the mitochondria, not the ribosome, we need the mitochondria, we need oxygen, we need enzymes, right, in order for that to occur. So other important coenzymes, which means these are special molecules that help drive metabolism, and that is NAD plus and FAD. Have you heard of niacin and riboflavin before in your vitamins or maybe the cereal box that you're eating? Yeah, niacin is where NAD plus comes from, and riboflavin is where FAD comes from. So these are special coenzymes that are involved. They pick up or drop off electrons depending where they're involved and what they're converted to when we look at metabolism. So let's start with the very beginning. So you've eaten your food, you've broken it down into glucose, and you have glucose freely floating in your blood. It leaves your blood and enters your tissues and enters your cell. But what is required to get that glucose in the cell? We're zooming out to the forest now. Think of just common sense stuff. What do you need to get your glucose out of your blood and into your cells? Because if you don't have this, you're called a diabetic because you can't get it out of your blood. It's stuck in your blood, can't go into your cells. What do you need? Insulin, yeah. So we're just going to take it at a grain of salt that insulin is present, and it's opening the door and letting that glucose into the cell. So once glucose enters the cell, it's phosphorylated. So what does that mean? It's phosphorylated. What do we say that term phosphorylation means? What's the key word in there? Phosphate. So what are we doing? Yeah, we're adding a phosphate. And where do we get the phosphate from? ATP, yeah. So we're kind of going in the wrong direction here. We're trying to make ATP from glucose, but we're using ATP to phosphorylate it. So do I have a diagram for this here? Yeah, that's coming up. So the first thing we have to do is change that molecule. Because how do things move through? Because glucose can enter our cells by facilitated diffusion. There's a couple of different ways. It also enters by secondary active transport. But if it's going to go by facilitated diffusion, there's no energy required for facilitated diffusion. But what is necessary to keep the flow going into the cell? Concentration gradient, very good. So it's got to be higher outside the cell than inside the cell. But we don't want that. We don't want to rely on a concentration gradient. We want to keep making ATP all the time. So what happens is when we convert that right away to something else by slapping some phosphates on it, it changes it from regular glucose to this glucose 6-phosphate. That keeps the concentration gradient and keeps glucose flowing into the cell. So by converting it to something else, it just gets it out of the way and allows for more flow into the cell. So some organs have enzymes that can reverse that, break off the phosphates, and let it flow out of the cell if we have low blood sugar. And that's the importance of the kidneys and the liver. They can release sugar to the blood if needed by reversing these reactions because they have the enzymes. So the only reason that the liver can do that is because it has the enzymes. And that's a test question, is why is the liver? Because it has the enzymes. So if we can't reverse the reactions because the enzymes aren't there in that cell. So anyway, that keeps the glucose concentration low, like I said, and ensures that glucose is going to continue into the cell following its concentration gradient. So when we talk about the whole process of cellular metabolism, like I said, we're oxidizing glucose. We keep removing hydrogens from that glucose. And then finally, all that's left is, how many hydrogens are in glucose to begin with? If you look at that reaction there. 12. And if I look at what's left if I take all the hydrogens off? If you look to the right of the arrow, what molecule remains when I remove all the hydrogens from it? We're looking just at glucose. What's left of glucose after I remove all the hydrogens off? Look at your choices. What else makes up glucose? Carbon and oxygen, right? If I take all these hydrogens off, look to the right. What's the remainder of my glucose? What's left? Yeah, carbon dioxide is what's left. If I take all the hydrogens off in a series of reactions, all I have left is carbon dioxide. but then I have a lot of ATP, so that's important. That's the goals. We're trying to get this storage molecule. We're trying to make fuel for our cells from glucose, because our cells can't use glucose for energy directly. We need ATP. So all that's left at the end of that is carbon dioxide, and that's a waste gas, right? Our bodies can't use carbon dioxide, so that's given off in our exhale. So the more metabolism we have going on, the more carbon dioxide we're producing, right? And then we have this water that's formed at the very end of the reaction in the electron transport chain. We have all these hydrogen atoms, and they bind with oxygen. Where do we get the oxygen from? When we breathe, right? Inhaling. So oxygen accepts hydrogen. So we're gaining hydrogen. What is that called when we gain hydrogen? Well, we oxidize the heck out of glucose, but what do we call it when we gain hydrogen? Yep, redox. So that's the reduction part. That's where those hydrogens end up. And then we have heat being given off, waste heat. Just like when you burn fuel in your gas tank, you have a lot of heat that's lost out of the exhaust pipe, right? So that's waste heat. And what do we call that waste heat? We measure it in our patients. Yeah, our body temperature, right? So your body temperature is a result of metabolism. And a lot of the metabolism that contributes to your body temperature is muscle metabolism, because the more we know that our muscles are part of that feedback system, right? That when we get cold, we contract our muscles and shiver to raise body temperature. Okay, so when we talk about taking this glucose and ending up with water, carbon dioxide, and lots of ATP, it requires 3 reactions working together in series, and that's glycolysis, the Krebs cycle, and the electron transport chain. So we're going to talk about each of these processes and how they contribute to these final products. So be sure you can rewrite this reaction, and what we're going to do in ALP is we're going to write this reaction like 10 times on a piece of paper, because repetition is the best way to remember it. And you can do that for yourself as well. So we'll start off with glycolysis. This is the first step. When glucose enters, like I said, we need to convert it to something else so it doesn't leave the cell, and we continue to have glucose entering. So there's 10 steps involved. We're not going to hold you accountable for all 10 steps, but you just need to know some basic things. And a really basic concept is that it's anaerobic. It does not require oxygen. So we don't need to be heavy breathing to make some ATP with the process that we call glycolysis. So this is important when people are doing really fast, intense exercise, like a sprint, like a 50-meter dash, or running up the steps, or lifting something really heavy and carrying it across your garage. Sometimes you're just going, ooh, right? And then you set it down. So you're not really breathing during that time, but you are using glucose, and it's not required for this short period of time. So it occurs in the cytoplasm. So this is not something that requires a mitochondria or an organelle. It occurs in the cytoplasm, and I want you to write after this statement, occurs in the cytosol. Why does it occur in the cytosol? Just like I said, why does it happen in the liver that we can release glucose? Enzymes, because the enzymes for glycolysis are present in the cytosol. So what happens then is we take glucose and we break it down into 2 pyruvic acid molecules. That's the end product. At the end of glycolysis, we know glycolysis is complete when we no longer have a sugar molecule, but we have 2 pyruvic acid molecules. So there's 3 phases of glycolysis. There's the activation, the cleavage, and then the oxidation and ATP formation. So we'll start with the first part, the activation. So remember I said we have to throw a phosphate on the end of that glucose molecule to keep it from leaving the cell and to keep the low concentration of glucose so we have steady flow into the cells. So we pop a phosphate on each side, and that we had to steal a phosphate from 2 different ATP molecules. So We bring the glucose in, and we use 2 ATPs to drop a phosphate on either side. So have you heard the phrase that you have to spend money to make money, right? Well, in some cases that's true, not always, but in some cases it's the same thing here, though. We're using 2 ATP to eventually make lots of ATP. So the process of glycolysis uses 2 ATP right off the bat. So we're kind of going in the wrong direction because the goal is to make lots of ATP. So this is called sugar activation. We're activating the sugar, getting it ready for glycolysis. So we add a phosphate to either end using 2 ATP, and we form 2 ADP from that, right? Because it went from triphosphate to diphosphate. All right, and then we cleave it. Then we split it in half. So we cleave those carbon atoms, split them in half. And then we have 2 different molecules here. So that's the cleavage part. Then, in the process of involving our coenzymes, NAD plus and NADH, or NAD plus is converted to NADH and H plus. So what happened? What happened to NAD? Did it gain or lose a hydrogen at the end of this reaction? It gained a hydrogen. Where did the hydrogen come from? When we split that sugar, right, when you split that newly formed phosphorylated sugar, we release hydrogen atoms, which then bonded to our NAD plus molecule. So all of a sudden now NADH plus H plus is formed. We've picked up energy, right? Because it gained an electron, because there's 1 electron in a hydrogen atom. So when it gains a hydrogen, it picks up energy. And in the process, we formed 4 ATP molecules. So we formed 4 ATP, and we have 2 pyruvic acid. What's the net ATP, though? Because remember, we used 2 to begin, so we have a net of 2. Even though we made 4 in this second process, in this oxidation ATP formation, we only have a net of 2. So at the end of glycolysis, we have 2 pyruvic acid molecules and 2 ATP molecules, net. And now we're going to go into the next part. Now, if oxygen is not available, it ends here as far as ATP production. So the next part then, the pyruvic acid will be converted in our muscles to lactic acid if there's no oxygen available, and we have just 2 ATP. So the bad part about glycolysis being anaerobic is that we don't get very much ATP formation, and the activities related to that, no oxygen activity, is very short-lived. It can be intense, which is good, like powerlifting or deadlifting can be intense activity, but it's very short-lived because there's not enough oxygen to fuel it. Or at least when you're done, you have to pay back your oxygen debt with heavy breathing. Like if you run up the steps without breathing, when you get to the top, you're breathing heavily, which helps convert the lactic acid back to pyruvic acid, and in some cases back to glucose. That's called oxygen debt. So the next part then is the Krebs cycle. So now we have these 2 pyruvic acid molecules, and if we have oxygen present, the pyruvic acid molecules are going to be converted to acetyl coenzyme A and begin the process of the Krebs cycle in the mitochondria. So this occurs in the mitochondrial matrix. We're going to call it the Krebs cycle. Some places or some textbooks, older textbooks, call it the citric acid cycle. But it occurs in the mitochondrial matrix. And I want you to know that the matrix is the fluid of the mitochondria. And why does it occur in the mitochondrial matrix? Very good, because the enzymes are there for that particular reaction. So it occurs in the matrix. It's fueled by pyruvic acid. And again, that's from glycolysis. So glycolysis has to happen first. But if we have fat only as our fuel source, it can also use fatty acids. So So that's where like the ketogenic diet works, right? Because we have other fuels to make ATP other than glucose. So pyruvic acid enters into what we call the transitional phase once it's in the mitochondria, because it needs to be converted to acetyl coenzyme A before it can enter into a series of reactions called Krebs cycle. So it's converted to acetyl coenzyme A, and then it enters this series of reactions. And I'm not going to expect you to know all the different acids formed along the way, but what I do want you to know is that these acids can, well, first of all, they release carbon dioxide. So if you notice, if you go kind of around the circle, you see we're removing carbon. And when we're removing carbon, breaking that bond, we're forming carbon dioxide, and hydrogen atoms are released. So again, do you see how NAD plus picks up more hydrogen here? So this is an oxidation reduction reaction, because this molecule, like if we just start out with citric acid, for example, and the next one, I can't even read what it says, isocitric acid. So we can see that that we're removing a carbon from it to form carbon dioxide. It's binding with oxygen. And then we're also removing hydrogens from it, which are added to NAD+. So if we're losing hydrogen, we're losing energy. So what's happening to this acid here? It's being, your choice is oxidized or reduced. It's losing energy, it's losing hydrogen, so it's being oxidized. And what's picking up the hydrogen atoms? NAD+. So NAD+ is being reduced, because remember, these reactions occur in pairs. So we're oxidizing these acids, and we're adding those atoms to NAD+. So, I mean, reducing, it's hard to stick that word, right, onto something that's gaining energy. It doesn't really, our brains don't want to make that association, but it's just a chemical term, oxidation reduction. So we're giving energy to this NAD+, because this is going to be important in the next step. So we have to get all these electrons built up on this NAD and FAD molecules. They're gaining hydrogen as well. Okay, so these are being reduced. These keto acids, you've heard of keto acids before, right? These are being oxidized. They're losing oxygen, or I'm sorry, losing hydrogens, forming carbon dioxide. So when you think of the Krebs cycle, what its function is to pick up electrons to provide energy for the electron transport chain, and carbon dioxide is produced as a waste product. So Krebs starts with a K sound, and carbon dioxide is a result of the Krebs cycle. So when I said make sure you know the products of cellular metabolism, carbon dioxide is from the Krebs cycle. That's where it came from. Okay, so then we go to the electron transport chain. And this is where oxygen is directly used to form water. So we've got to have oxygen. Well, we have to have oxygen for the Krebs cycle as well, but it's not directly used. We can see here that it binds with the carbon taken from the acid here to form carbon dioxide. So it's not used, but it's part of the process of carbon dioxide formation. Okay, so anyway, so we have these coenzymes, NADH and H+ and FADH2, that were formed in the Krebs cycle, right? They picked up those hydrogens from these keto acids, so this is where they were formed. And then they use those to fuel proteins in the electron transport chain. So we'll look and see how that happens. And then some of those hydrogens are also going to combine with oxygen to form water. So if I look at this diagram, where does the electron transport chain occur? Did I say that? Did we miss that in my slide? Well, where it occurs is in the mitochondrial membrane, which is called the cristae. It's spelled C-R-I-S-T-A-E, cristae. and that's the membrane of the mitochondria. So this is where we have all these proteins embedded. So here's the mitochondrial outer membrane. Here's the matrix, the fluid, right? And then here's the inner membrane of the mitochondria, which we call the cristae. So we can see there's a number of proteins embedded there. And then we have this special protein. This is the end goal. This is what we're fueling. This is the Oz of cellular metabolism. And this is the ATP synthase pump. So the goal is to take these coenzymes, NADH plus and FADH2, or NADH and H plus and FADH2, is those electrons then are released as a result of these chemical reactions with these proteins embedded in the mitochondrial cristae, and hydrogen ions are pumped out into the matrix and serves as a fuel to drive this pump and to get it to cycle. And as it cycles, it phosphorylates an ADP to make ATP. So the hydrogen atoms are the fuel for the ATP synthase pump. So we have a lot of hydrogen atoms being released from those coenzymes, NADH and FADH2. Some fuel the pump, and the rest combine with oxygen from our inhale to form H2O. So I have an animation here that shows these processes, but let's just kind of summarize the key events. So the electron transport chain. We have energy from electrons from NADH and FADH2 that are pumped to the inner membrane. This is the mitochondrial, right? We're in the mitochondria, creating a proton gradient for H+. So that's this phase 1 is referring to this process right here, where we're pumping hydrogen atoms out into this space right here to make a proton gradient, which is H+, which is going to drive our ATP synthase pump. All right, then the proton gradient travels through the protein, the ATP synthase pump, making ATP. We call it chemiosmosis because we're using protons to flow through the ATP synthase pump from high to low concentration, just like we see water flow through aquaporins, through protein channels from high to low concentration. So this is kind of a close-up of the ATP synthase pump. It's a really cool looking pump, and I think I could include a picture of it. Yeah, on the next slide. So you don't have to know the details of the stator and the rotor and all that, but I just want you to know that hydrogen atoms, or hydrogen ions, that have lost an electron, so these are positively charged, these are cations, flow through the pump and drive the process of adding the phosphate to the ATP. So if we look at a cell membrane, a mitochondrial membrane, the cristae, this is what it looks like. Look at how many there are. It's just crazy. A lot of ATP production in our cells. So at the end of that, 1 glucose can make up to 2 in glycolysis. When we look back at the Krebs cycle, how many ATP did we get? Well, we had 1 ATP per pyruvic acid. How many pyruvic acid molecules were created in the Krebs cycle? There were 2. So at the end of the Krebs cycle, we process each of those pyruvic acids, we get 2 ATP. How many do we get from glucose? 2. And then when we look at what we could potentially get from the electron transport chain, it can be about 28. So if we add them all together, go down to our next slide, we get 2 from glycolysis, a net of 2, right? Because we actually produced 4, but we needed 2 to start things off. So we have a net of 2, 2 in the citric acid or Krebs cycle, and about 28 from electron transport chain. We can get up to 32. It says 2 ATP were lost in the process of shuttling ATP to the source of or the cell that's going to use it, but we end up with 30. It can be as high as 36 in some cells. Like kidney cells are really busy cells, constantly filtering our blood, so they use a little more, they can produce a little more ATP. But it's all about, isn't it, really, it's all about the electron transport chain and having that oxygen available. But we need oxygen for the citric acid cycle because that's what the carbon bonds to, right, for carbon dioxide. But we do not need oxygen for glycolysis. That's the only anaerobic process in cellular metabolism. Okay, so I'm going to show you a quick animation here of cellular respiration.
How do you turn that bite of food into a chemical that a cell can recognize and use as energy? The first step is altering the food into its component chemical compounds and then getting those molecules into your cells. That process is called digestion. Once inside your cells, the process of turning that bite of food into useful energy by cellular respiration begins. The process of digestion results with carbohydrates and other molecules being removed from the consumed food and transported into the bloodstream. From there, nutrients, like the carbohydrate glucose, will leave the bloodstream through a capillary wall and enter a tissue cell. Once inside the cell, cellular respiration will completely oxidize the glucose molecule, releasing high-energy electrons. The overall goal is to make ATP, a storage form of energy for most cells. Cellular respiration is a 4-stage process that begins with glycolysis. Glycolysis literally means splitting sugars, and it is the first step of cellular respiration, occurring in the cytoplasm of the cell. Glycolysis consists of 2 distinct phases, an energy investment phase and an energy harvesting phase. In the energy investment phase, 2 ATP molecules transfer energy to the glucose molecule, forming a 6-carbon sugar diphosphate molecule. This molecule splits, and the energy harvesting phase begins. During this phase, the 2 3-carbon molecules are converted to pyruvate, and ATP is formed. Glycolysis is a 10-step reaction that involves the activity of multiple enzymes and enzyme assistants.
I should mention pyruvate and pyruvic acid are the same thing. It's 2 different ways of saying it. Okay, so when you hear pyruvate, pyruvic acid.
Process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins. The next stage of cellular respiration involves the movement of pyruvate
So going back to, oops, I lost my PowerPoint. Here we go. Okay, so applying this knowledge of metabolism then to what we know, one piece of information is the efficiency of this process of, and you should write this down, of the energy that's used, or of the energy that's in the bonds of glucose. So glucose has energy stored in its chemical bonds. Through the breaking of those bonds and converting to ATP, what we get, the energy, the transfer, we only get 38% of the energy out of glucose into ATP, 38%. So where does the other 62% go? Just like in your engines, where did I say the other 32 or the other 62% of that energy from glucose, where did it go? Yep, off as heat. So 38% we actually get to utilize in the form of ATP. So oxygen is required to fuel metabolism, right? To keep our cells doing what they need to do. So here's another take-home point. What do we care about all this stuff? Well, we know we need glucose, right, to fuel metabolism. We know, and where does the glucose come from? Where did it all start? Digestion. Okay, so our patients need to be eating, right? And if they're not eating, the body will get glucose by burning protein and fat. Okay, so it needs to be in the blood. We need to have sugar in the blood to fuel metabolism. So if your patient is lethargic and unresponsive, the first thing that we can do quickly, we have to call the doctor, right? But the first thing we can do quickly is check their blood sugar. Yeah, check their blood sugar. So that would be the first thing you're going to do is get a blood sugar. The next thing you're going to do, which is quickly, you're going to do these all simultaneously. So you would, as a nurse, you would, like at Gunderson, we don't do blood sugars as nurses unless we work on pediatrics or certain specialized areas. I think charge nurses can do blood sugars, but typically only the CNAs have regular certification for checking blood sugars. So you'd tell your CNA, let's get a blood sugar, and then you would check what else fuels metabolism. Oxygen. So you're going to check their oxygen levels. How do you check their oxygen levels? Yeah, O2 sat on the fingertip, right? So you would check that. And then if that's low, what can you quickly adjust? Yeah, oxygen on the wall, supplemental oxygens, turn that up, right? So fuel metabolism that way. Another thing that we're going to learn later in the semester is that if carbon dioxide is building up in the blood and they have a lung issue, that their lungs don't allow carbon dioxide to diffuse from the blood into the lungs to be exhaled out, and it builds up in the blood. These acids that are formed from the Krebs cycle, this carbon dioxide, along with the keto acids, causes a dangerous pH drop. And that can cause people to be really lethargic because the enzymes aren't working well because their blood pH is too low. Because enzymes depend on just the right conditions. Temperature has to be right, pH has to be right in order for enzymes to work properly. So if pH drops because of too much carbon dioxide in the blood, because carbon dioxide, we're going to learn later, can form carbonic acid, and which can break up into ions and release hydrogen ion, and we know more hydrogen ion drops pH. So we'll sometimes have to do blood gases, but that's a more detailed thing. And by that time, your doctor is there, and they'll order the blood gas. But things that you need to think about are blood sugar and O2 sats. That's something you can check quickly. And what did we say happens if there's not enough oxygen? Let's say you have a patient and he has a narrowed vessels over the heart muscle, so there's not enough blood flow to the heart muscle, so the heart muscle cells aren't getting enough oxygen. What pathway are they going to use to get ATP so the heart keeps beating? The anaerobic pathway. So of the three processes we talked about, glycolysis, Krebs, and electron transport chain, which of those three will it use? Glycolysis only. And then when glycolysis produces the pyruvic acid, what is that converted to in the cytoplasm? Did we say by the process of fermentation? Lactic acid. So and the heart is a muscle, right? So when it takes pyruvic acid and breaks it down into lactic acid, how do your muscles feel when you climb up the steps and you're doing intense anaerobic activity? What do they do at the end of the activity? Or what causes you to stop? Do you feel the burn? Yeah, the burn. Well, that's the same thing that's happening in patients that have a narrowing vessel. They're outside mowing the lawn. Their heart is needing more oxygen. It's not getting it. It's converting to the anaerobic pathway. It's producing lactic acid in those heart muscle cells, and that burns. Sometimes it feels like pressure in the chest. And sometimes the way the nerves fuel the heart or serve the heart, that can cause pain in the jaw on the left side, or even both sides. Can cause pain in the teeth, can go up to the ear, and down the left arm. So whenever patients complain about any of those kind of pains of pressure, discomfort, burning, that's what we call angina, and that's a sign of heart muscle cells are not getting the oxygen they need for metabolism, and they're making lactic acid. And we need to correct that quickly. So what's the first thing you can do? It's all about a lack of what? Oxygen. So what's the first thing you can do when a patient says they have chest pain? Give them some oxygen. Crank up the oxygen. See if that helps. And then the doctors will come. And then patients that have heart issues and they know they have narrowed vessels, what do they carry around in their pocket or purse? Nitro. Because nitro, when we put it under the tongue and let it dissolve quickly into the mucous membrane, goes into the blood, it opens up vessels. So if it opens up narrowed vessels, it's going to increase blood flow. But we've had patients, they come in and we put them on a nitro drip to open up their vessels. So it's going in via IV and they feel great. Like, I feel really good. I'm ready to go home. Well, we have to tell them, you can't take that IV pole home with you and be on nitro all the time, keeping your vessels open. We have to correct the problem, and sometimes that's bypass surgery, sometimes it's a stent, or sometimes it's just a medicine that helps dilate the vessels. So there's things we can do. But understanding the role of metabolism is anytime a patient has increased metabolism, they're going to have increased oxygen needs. So what are some things that can cause increased metabolism in a hospitalized patient? What? Okay, if they're having a hard time breathing, they're using a lot of muscles to get air in, that's going to burn more oxygen, isn't it? So when are they going to need to breathe more? What type of activities are they needing to breathe more? Going to therapy? Getting dressed, getting cleaned up for the day, right? Going into the bathroom. So those are all things that are going to require increased metabolism, increased oxygen needs. So we need to maybe sometimes crank up the oxygen if they're going to go and perform certain activities. What about if a patient's having seizures? Is that going to use more metabolism? Could that drop oxygen levels? Definitely. So when you talk about the old wives' tales, feed a fever, starve a cold. Feed a fever, would a person with a fever have more oxygen demands? Yeah, because what is the increased temperature? That's part of the immune system, right? But oftentimes if you listen to the heart rate of a person with a fever, their heart is beating faster because the body is burning more fuel. So therefore we do need to feed people that have fevers, encourage them to eat, right? Because higher metabolic needs. So we're going to continue this conversation in the next set of the notes where we talk about how we convert different food products to fuel metabolism.